All right, we will go ahead and get started. There are several people on Zoom as well. Tis the season for colds, COVID, and other maladies. So I think we're trying to stop this from being a super spreader event. Okay, so first of all, welcome to the last Pioneers in Biomedical Research Science, Biomedical Research seminar for the calendar year of 2023 and for the semester. I want to remind you that in 2024, starting on January 12th, the series resumes with Ben Garcia from Washington University and followed in the next 2 weeks by Ondine Cleaver from UT Southwestern and then Marnie Halpern from Dartmouth. So today it is my pleasure to introduce Tim Petros, who has come to us from the National Institute of Child Health and Human Development at NIH in Bethesda. Tim is, both part of my broader scientific family because he was a graduate student with my friend Carol Mason, who I worked in the lab that I worked in as an undergraduate. So there's a connection. But Tim has a distinguished academic history and scientific record of accomplishment on his own. He was an undergraduate at Brown University, where he began his journey through neuroscience. He worked with J.D. Miller. He actually was involved in a publication on pain mechanisms. He then went to Columbia University as a graduate student, where he worked with Carol Mason on the molecular mechanisms of axon guidance and axon crossing at the optic chiasm, and was involved in some of the really pioneering work working out the molecular mechanisms of that very important example of axon guidance and choice. He then went from Carol's lab to Stuart Anderson's lab at Cornell University, where he began his focus on one step back in developmental neuroscience on progenitor and stem cells, particularly for interneurons, which is an interest that he's retained. And with Stuart, looked at some of the mechanisms and secreted factors that influenced interneuron progenitor progression and differentiation. And then he moved to Gord Fischel's lab, where he continued this interest with Gord at NYU. In 2017, he accepted a position at the National Institute of Child Health and Human Development as an investigator, where he began his own lab and really began to do really elegant pioneering work on the epigenetic regulation of progenitor cells in the nervous system. And I think this was really a bold move because Tim's work has really focused on looking at these issues in the actual progenitors from the actual developing nervous system, which is a challenge in and of itself. And he has really provided some really foundational, elegant evidence of how these things might work. And so it is fitting that he should be called a pioneer. He did not come in a Conestoga wagon. We will not make him wear a bonnet. However, it is a real pleasure to welcome him here today. So, Tim.
Okay. Thanks, Anthony, for that kind introduction and for inviting me here to talk about my lab's work. It was a pleasure meeting with some of you today. I look forward to meeting more of you this afternoon during my discussion. As Anthony mentioned, okay, perfect. My lab investigates the mechanisms, both genetic and epigenetic, that regulate fate and maturation of forebrain GABAergic inhibitory interneurons. Now, I think most people in the audience are probably familiar with brain development, but over time, you go from the small tube of cells to a much more complex gyrated structure in humans. And how you go from this structure to this structure, I think, is one of the most challenging and exciting questions in basic biology. A lot of us, it's very complex to study the human brain, so a lot of us use smaller model organisms. The mouse up here, this is all drawn to scale. both in a lateral view and a cross-section view. So even though the mouse brain pales in comparison to the human brain, it is still extremely complex with billions of neurons forming trillions of synapses. So how you generate this diversity is just an incredibly exciting and challenging question to undertake. So the cells we study are these GABAergic interneurons highlighted here in green. They're born in the ventral part of the brain. This is a cross-section through an E13 mouse brain. There's these ganglionic eminences, these bumps in the brain, the lateral and the medial, and not pictured here is the caudal. These cells are born in the medial and caudal ganglionic eminence and then migrate throughout the brain to populate every brain region where they perform critical functions in nearly every aspect of brain circuitry. Now, we know a little bit about some of the mechanisms that give rise to these. Work over the years has identified some genes that are critical for generating MG and CG-derived interneurons. Additionally, different cohorts of mature interneurons arise from these regions. So MG gives rise to parvalbumin and somatostatin-expressing interneurons, whereas CG gives rise to CCIP and other cohorts of interneurons. When you get to the adult mouse brain, these cells have migrated and populated all of the regions shown here. And just to give you a feel for the diversity, if you zoom in, looking at real reconstructions here on the left or a schematic here, you see the very sad excitatory cell in gray with all the different diverse inhibitory cells that can synapse on these cells. In addition to being categorized based on physiological properties and morphology, the advent of single-cell sequencing now allows us to look at their gene expression in the transcriptome. And so despite the fact that interneurons only make up about 20 to 30% of the cells in the brain, they actually occupy about 50% of the genetically defined cell types in the brain in mouse. And this even expands to closer to 2/3 in the human brain. So it just highlights kind of the diversity of these cell types that play different roles in various aspects of circuit function. Now, it goes without saying that perturbation or dysfunction of interneurons are related to a wide variety of diseases, developmental diseases, some of which are described here, but there's plenty of other ones. Two other kind of important notes is that many genes associated with these diseases are enriched in neuroprogenitors, so cells within the embryo or the early stages of neurogenesis. And in some cases, some of the genes are actually enriched particularly in immature interneurons. And second is that there's growing evidence from GWAS studies and other types of approaches that mutations in proteins that regulate epigenetic machinery are often associated with psychiatric disorders. So this has kind of led to an overall theme in our lab where we believe that we really need a better understanding of both the genetic and the epigenetic processes that regulate fate and maturation of these inhibitory interneurons in order to understand both normal development as well as to have insight into disease etiologies. So I'm going to take you through a couple of stories today. The first is that in order to get a handle on this, we kind of needed to know the ground state of how these cells look in these different brain environments as they're making these critical decisions, initial decisions in cell fate. And so to do that, we took a multimodal approach. We performed single cell sequencing, single cell ATAC sequencing to look at the transcriptome, the chromatin accessibility. We also wanted to assess histone modification so that we had further insight into whether a region was an active promoter, an active enhancer, or repressed. And we also wanted to look at higher order chromatin structure in the brain to compare and contrast differences between these brain regions. And only when you have this kind of multimodal data set can you really kind of understand and start to dig into how these cells make these decisions in the embryo. So once we establish this kind of ground state, then we currently, one study is finished and one is ongoing to look at what happens in the MG in particular when you perturb either gene repression or gene activation. So basically kind of taking a sledgehammer to manipulate gene expression and see how that affects cell decisions, cell fate, and ultimately cell behavior. So we do a lot of sequencing in the lab, and this is kind of highlights the general workflow where we can take our mouse embryo at whatever age we prefer. It'd be lovely if the brain was actually stained brown, but it's not. But you can still dissect the regions we care about. You can see them anatomically, the medial, the lateral, and the caudal ganglionic eminence, and the cortex. So we can dissect out these regions, dissociate them to obtain either single cell or single nuclei dissociations, and then put them through the 10x pipeline on our lab to do single cell sequencing. or collect the tissue after FACS for higher order bulk sequencing for some of the other approaches. Send it to the sequencing court and ICHD, and then what we spend most of our time doing is a computational analysis, figuring out how to mine this data to figure out what it says and what it doesn't say. And depending on the day, my postdocs feel like kissing their computer or smashing their computer, depending on how many errors they get trying to run their bioinformatic analysis. So multiple labs had kind of already done some single cell RNA sequencing when we got into this work 4 or 5 years ago. But looking at chromatin accessibility to get further insight into enhancers or promoters was really critical. And so kind of when we were starting this idea, there were ways to look at chromatin accessibility called ATAC-seq, which I'll show in a minute. But if you have a heterogeneous cell population like we do in the developing brain, if you sequence the accessibility, you get all these hits where regions could be enhancers or promoters, but you don't have the specificity you need to look at differences. So at the advent of single cell ATAC sequencing, now you can either FACS sort or genetically identify the cell types you want to really get some insight and to look into how chromatin accessibility is different in distinct cells in a heterogeneous population. So the way this assay works, it's an assay for transposase accessible chromatin sequencing. Basically, you have the DNA with a transposase that can go in and insert itself into open regions of the chromatin. It's been modified to add adapters. It can cut the DNA, add in these adapters, and then you harvest these small regions and sequence it. And that tells you regions in the genome that are open or accessible to translation or transcription or possible enhancers. So this is just kind of the pipeline. I sort of showed this already. Dissect the different regions, pump it through the sequencing. Any hits, any regions where you see lots of hits will register above a threshold, and then you call that a peak. So a region is accessible in that genomic loci. What you're looking at here is a UMAP plot where each dot represents a cell. So 2 dots that are very close together have very similar chromatin accessibility profiles, and 2 cells that are very far apart have very distinct chromatin profiles. And you can see that the cortical cells, the excitatory cells are all kind of clustered together over here. The MGE cells over here and the LG and CG, for which there's less known genetic differences, are kind of intermingled with each other in this region. So this looks very similar to a type of single cell RNA-seq you would look at. You can divide these cells into putative cell clusters. We can group them based on whether they're apical progenitors, ventricular zone cells, basal progenitors, or neurons, and you can perform pseudotime based on gene expression profiles. Now, we can also look at the accessibility of specific promoters. So just here's some examples. Nestin is a gene enriched in radioglial cells. And you can see that the promoter is only accessible in the radioglial cells down here and becomes inaccessible or repressed over time. And we can see that type of accessibility profile change over time as well as over space. NeuroD6 is a gene restricted to cortical cells, excitatory cells, and that motor is only accessible in NeuroD6. So you can investigate particular genes of interest in this data set. You can also look at the accessibility profile looking on the putative clusters. So here's NKX2.1, a gene that's really only expressed in the MGE, and you can see that it's only accessible in the clusters from the MG, and it's closed off in these other regions. So there's lots of different ways to kind of look at this data. Another thing we did that was a little bit more unbiased is to look at differential accessible peaks between regions. So in this case, you have all the 24, 25 putative cell clusters that we have here, color-coded by which tissue they came from or which cell type they are. And basically, you can kind of compare each cell cluster with each other to look at the differentially accessible peaks between them. So here you have 4 different clusters that are all from the CG, all from the cortex and all ventricular zone cells, and they have more similar accessibility profiles to each other than they do to other regions. And then what's also nice is within these open chromatin regions, you can look for transcription factor binding sites within these accessible regions. And we saw a lot of binding sites that are related to genes specifically expressed in the cell type. So EMX1 is a cortex-specific gene. DLX and LHX6 are GABAergic cell specific genes. So we see transcription factor binding sites restricted to open regions of the chromatin that are consistent with the gene expression profiles. Now, we also performed single-cell RNA-seq. This was before the multiome existed, where you can do the same thing in both cells. So in a previous study, we performed single-cell RNA-seq, and this is just one example where, in this case, we're highlighting the ventricular zone cells. We see significant heterogeneity and gene expression in these ventricular zone cells. And then we followed it up to look at differential patterning. We find various genes are enriched either in the caudal or dorsal or ventral or dorsal regions of the CG. And so there's a lot of spatial information you can get even from our single cell sequencing. So we basically had this single cell RNA transcriptome data and single cell ATAC data to look at chromatin accessibility. And Chris Rhodes, my former postdoc, was able to integrate this data together in the same computational space. So even though these are different data sets, nowadays these would be the same cells, but in this data set, they're different cells. You can integrate them and see how the RNA level changes with the chromatin accessibility. And so what we found is here's a cohort of genes whose expression was high early on in pseudotime. So this is in the progenitor cells, it's enriched. And then the expression of these genes go down as these cells become postmitotic. And a trend we saw is that the RNA here in blue tended to decrease quicker than the GAS is the promoter accessibility and enhancer promoter. The accessibility of these regions seemed to decrease after we saw RNA levels going down. So this implied kind of a relationship between RNA and chromatin accessibility that was somewhat surprising. Conversely, there's other genes that go up over time, and you can see that both the promoter and enhancer accessibility increases prior to RNA turning on. So this just gives a little bit of a feel how you can look at chromatin accessibility and transcriptome data together. Now, this is great, but when you look at accessibility, you really don't know what those regions are. You just know if they're open and closed. And so one way to try to gain additional insight onto that is to look at the histone modifications that can really provide further information whether a region is an enhancer or promoter. And so there's lots of different histone modifications that are associated with this. There's 3 in particular that are indicative of an active promoter, an active enhancer, or a repressed region. And so we wanted to see how these histone modifications were different between these distinct regions in the embryonic mouse brain. So we performed cut and tag to do this. This is kind of a fancier, cheaper version of ChIP-seq to look at these histone modifications. And so now we have both gene expression data for single-cell RNA, chromatin accessibility, and based on histone marks, active regions and repressed regions from the 4 different brain regions. And in general, we saw a lot of similarities, but I do want to point out, if you look at this gene here, NR2F2, that's really only expressed in the CGE. If you look at the chromatin accessibility, the promoter is actually wide open in all 4 tissues, despite the fact it's only expressed in the CG. So this I just like to highlight because you really need this multimodal approach. If you only look at one aspect of this, either RNA or accessibility, you could be misled, such as here, you would think that this gene is probably expressed in all of these regions. So only by having all of this information integrated together can you really have a good sense and understanding of what's going on in these cells or in these brain regions. Now, if you look at the mark for active promoters, I'll just highlight one gene here. This is ASCL1. It's a gene that's expressed in these GABAergic cells. It's not expressed in the cortex. And so up here you have the accessibility in the 4 regions and activation. I just want to highlight this region right here. This was a region thought to be an enhancer for ASCL1, and they made mice for it, and it didn't really replicate the expression pattern ASCL1. If you look at our data, you can see that that region is accessible in all 4 brain regions, and it does have an active mark as an active enhancer, but the actual strongest enhancer mark is in the brain region, the cortex, where this gene is not expressed. So we'd actually argue that this is probably not a good candidate as an enhancer for this gene because the strongest signal is in a region where it's not expressed. Conversely, if you look just downstream, you see this loci here, where there's high indicators of active enhancers in the 3 brain regions where it is expressed, and it's much lower here. Same thing looking at accessibility. So we'd argue that this loci here that hasn't been studied is a much stronger candidate for a potential enhancer of ASCL1 activation. And so we've found similar regions and other genes that we think provide some insight into high confidence enhancers at genes that play critical roles in neurodevelopment. Now, the last piece of data we wanted to get was to look at higher order chromatin structure. And so just to kind of give a brief review of this, the genome is actually highly organized in these topographically associated domains or TADs. The way this works is that you have CTCFs and a set of other proteins that basically sequester genomic regions such that DNA over here can only interact with itself and not interact with regions over here. And that's how you can have localized promoter enhancer networks. And there's all kinds of different versions of this. You can get these massive kind of promoter interactions where you have strong upregulation of a cohort of genes. And so one way to analyze this is to perform Hi-C, which basically you fix tissue, which sort of glues the genome together so that adjacent regions are now basically attached. You then can fragment the chromatin, put adapters on here, glue these adapters back together, and now you have a library of DNA with two adjacent pieces of chromatin that tells you that this purple and this orange region were in contact, and again, gives higher confidence for important interaction. And so if you haven't seen this type of data before, this is how Hi-C data is typically represented. You see kind of these hotspots here and here, and this is a point of genomic interaction. And so the way you interpret this is it's like a pyramid. You follow it down the slope this way. And when you come to the DNA loci down here, this hotspot means that there's an interaction between this region of the DNA and this region of the DNA here. And so what we found is that if we look at particular genes, so this is NKX 2.1, again, an MG restricted gene, we performed Hi-C on these 4 different regions. What I hope you can appreciate is that right here, you kind of see this hotspot of NKX 2.1 of an interaction of the NKX 2.1 promoter that's present in the MGE, but absent in the LG and the CG and the cortex. Conversely, over here, you see some hotspots in these 2 regions, in these 3 non-MGE regions that seems to be absent in the MGE. And if you blow this up and look at Capture-C, which is sort of a modified version of Hi-C, you can see it confirms what we observe, where you have a specific interaction at the NKX 2.1 promoter, which is right here, with this region downstream that's present in the MGE, but not present in the other regions. Conversely, on the other side, you have these interactions in these non-MGE regions with the NKX 2.1 promoter that's absent in the MGE. And then you can fill in the other data we have to look at the RNA and the histone modifications. And so what this nicely shows is that in 2 brain regions, side by side, you have completely different chromatin organization at a gene that's critical for fate and maturation of. Yeah. So you can have both. What I blew up here, all we're looking at is a region of the chromosome where NKX 2.1 is, but you can look at all 22 chromosomes at the same time, and you are able to see different chromosome interactions. That's much more rare. You need about 5 million cells to do this and sequence it at an absurdly high depth. So it's really hard or rare to get different chromosome interactions, but you can look at that in this type of data set. And what's interesting is that, so where this interaction occurs is at this gene MBIP. MBIP, we found, this name might look familiar, is also strongly enriched in the MG. It doesn't seem to be expressed in the LG or the CG. So here we have an interaction specifically of the MGE for 2 genes that are only expressed in the MG. And so rather than an enhancer-promoter interaction, this might be a sort of promoter-promoter interaction where they're both helping each other's expression in the MG or restricting it elsewhere. And again, we've found other intriguing regions where the chromatin interaction is distinct in these brain regions that give rise to different types of neurons. So to go back to the region that NKX 2.1, we worked with our colleague Pedro Roca, where he's designed a creative strategy to try to perturb interactions, genomic interactions, without disrupting coding regions. So here's NKX 2.1 here, the MBIP gene is here. You can look at these CTCF sites, and basically he's designed to insert some CTF sites in here to basically make an artificial barrier so that these 2 regions cannot contact each other. So in the normal MG, you see this type of relationship. And by inserting these CTCF regions in here, the idea is to insert a barrier where these two regions can no longer interact. And so we've generated these mice from the preliminary data that we have. The good old N of 1 shows that basically in this mutation where we've theoretically disrupted this interaction, expression of MBIP is basically lost in the MG. So this shows that by perturbing this promoter enhancer interaction without touching the gene regulatory, the gene coding cassettes, we can alter expression of this gene. And so we're currently still working on this to figure out the role of MBIP in this region and if NKX 2.1 is affected, and also cell fate. So this is a ton of data we generated, and it's really important for us to make it as accessible and usable as possible to the community. So we've imported all of our data into the UCSC browser, so anybody can go in, type in their favorite gene, see the RNA data, the accessibility data, co-accessibility, which I didn't even go into, all of the histone tags to look at putative active enhancers, promoters, and then in the end, the Hi-C data. And so we think this is a really powerful resource for anybody looking at the embryonic mouse brain to really understand, compare and contrast these different brain regions, both genetically and epigenetically. Okay, so in conclusion for this part, we've used a combinatorial approach to look at different genetic and epigenetic status of these cells in the embryonic mouse brain in these 4 distinct brain regions that give rise to distinct cell types. And again, we just highlight that each data point is just one piece of the puzzle in order to really understand how genes and epigenetic mechanisms play a role in cell fate. You really have to kind of put all of this together to really understand the story. We think we've identified a number of high confidence enhancers at various genes that are critical for cell fate decisions at this time that we think are worth studying, and some of them we are following up on. And what was really exciting for us is to see this distinct chromatin architecture within the brain, because this has not been well explored, or I'm not sure well studied at all. And it really tells about the different higher order structure that is setting up these different regions to be able to generate these different and distinct cell types. And we really think investigating chromatin structure is a huge missing piece of the puzzle to really understand neurodevelopment. So now that we had this data set in hand, we were well positioned to figure out what goes wrong in gene expression and how that affects these various aspects. Yep.
The region that you're focusing on looks like it's part of a larger region that's present in all your brain regions, like a larger associated domain. Yeah, like that is part of a bigger region. Are those just that are that's expressed in all your different brain regions? So I don't know if it's this one, but like if you were you focused on it the first time?
Zoomed in. This?
Yeah, like it's like the bigger region, the bigger pyramid region that is in all of your different brain regions where the left line is.
Yeah.
Is that just, are those just like things that are generally expressed in the brain within that?
I mean, presumably, so we haven't followed. So here's an example where you have this hotspot kind of in forward. So if you follow this down, so the NKX 2.1 promoter is kind of over here. So it's quite possible this is an interaction with the NKX 2.1 promoter here with a region somewhere over here. But this is present in kind of all 4 brain regions, so it was of less interest to us. But yeah, so this would indicate an interaction that's common in the regions. And most, I looked at about 50 or so genes. The majority of them, there wasn't anything vastly different or exciting between it, but there's a handful where it was, and it tended to be with genes that we know, transcription factors that we know play critical roles in fate of those cells. And also, so this resolution is good, but not great. The resolution is here. You're talking about many kilobases where this interaction is. There's a newer technique that we're now trying now with Petros that's called micro-C that really has a resolution of tens of nucleotides. So with there, you really have the resolution to have way more confidence that this region of 10, 20, 50 nucleotides is directly interacting with the promoter. So we'll have much higher resolution in the future and be able to maybe address some questions in more detail. Okay, So, so our first approach was to kind of take a sledgehammer to the system. What happens if we massively perturb gene repression or gene activation? How does that affect cell fate rather than looking at kind of the function of a specific gene? And so my former postdoc had actually in his doctoral training had worked with this gene EZH2, looking at its role in monkey and some other stuff. But what was relevant for us is that it's a component of this complex called PRC2 that's really critical for gene repression. So it's involved in H3K27 trimethylation. When this residue gets methylated, that's critical for gene repression. Other labs had investigated the role of EZH2 in the brain. It's highly studied in cancer. Its activity is often modified in a lot of types of cancer. In the brain, there's only been a handful of studies that had looked at it, but in general, if you perturb EZH2, you see changes in cell fate, and cell migration, and you can kind of sometimes see a shift in fate and how cells respond to cues. And these are all important aspects for interneuron development. They have to migrate a very far away. So any perturbation and cell cycle dynamics and migration was intriguing to us. So we wanted to explore what happens when you knock out EZH2 in the MGE. So the first thing we did, imported the conditional knockout, crossed with that case 2.1. You can see strong downregulation of EZH2 in the knockout. And then if you look at the readout, the K27 trimethylation, again, you see a strong reduction of the K27 trimethylation in the MG in the knockout. So it looks like we have perturbed methylation and thus gene repression in this mouse model. Now, when we look at in a P21 cortex and we look at the presence of different interneuron subtypes, so this is crossed with AI9, so all MGE-derived interneurons are in red, and then the two main subcategories, somatostatin and parvalbumin. What we saw is, as a general, a slight decrease in the total amount of MG-derived interneurons in this mouse, but we actually saw a shift in the fate. So we saw an increase in somatostatin cells and a decrease in parvalbumin cells. And that was true whether you look at overall density or the percentage of these cells. So you're making more of one cell type at the expense of another in this mouse line. We also perform cell counts in the striatum and hippocampus, and we see similar changes in the differences in fate of these interneurons in this mouse. I'll also note that when we use a different Cree driver line that specifically knocks out the gene in post-mitotic cells, we do not see these changes. So the function of EZH2 is really critical in these cycling cells, where loss of the cycling cells has this strong phenotype, but loss in the post-mitotic cells does not. Now, because we see these changes in the adult mouse, we wanted to look at an earlier stage. So we looked at P6. Unfortunately, we can't let parvalbumin doesn't start to be expressed until about P10 and then ramps up. So we can't really look at the specific subsets here, but we do see this strong decrease in MG-derived cells present at P6. So we're quite confident this is changes in cell cycle dynamics and neurogenesis. These cells are being kicked out of the cell cycle too early and depleting the progenitor pool, and that's leading to a decrease in cell numbers, and in particular, a decrease in the percentage of parvalbumin cell numbers. Now we wanted to see how happy are these cells that survive? Do they have the normal physiological characteristics of interneurons? So we collaborated with Soo-Yun Lee at NIMH, who was also with a postdoc in Gord's lab with me. And her postdoc, Siobhan Naskar, performed recordings from the cells in the mutant. And what they found is basically there's no physiological differences or EFIS changes in any of the intrinsic properties in these cells in the mutant compared to the wild type. What they did find, however, is changes in the morphology. It doesn't come out too good here, but they saw increased arborization of the axons in fast spiking cells or parvalbumin cells in the mutant compared to the wild type. And here's the shoal analysis there. And so we think this could be potentially a compensation. So you have fewer PV cells in the mutant mouse brain. Well, how does the brain try to compensate for fewer PV cells? One possible way is that the PV cells that survive have longer axons, more synaptic input to try to ramp up the inhibition. So we think this could be an example of compensation. Yep. So this was a somatosensory cortex, so they're recording from primarily layer 2, 3. But yeah, so that's they haven't looked at any other brain regions.
Yeah, or even for the neuron, the density, does that matter? Have you looked at other cortical areas? Or?
Looking at the cortical areas in terms of cell counts or to the physiology.
Well, either, I mean, electrophysiology is probably harder because it's more labor intensive. But
Yeah.
so-
So electrophysiology was only somatosensory cortex. Most of our cell counts were also somatosensory cortex. We did perform some, I think, in visual cortex or somewhere further away, and the trends were similar. We didn't do as careful and detailed analysis as we did compared to somatosensory, but it didn't look like there was shifting of cells, like a migration problem. So a lack of cells in the somatosensory because more were in the motor visual system. It didn't seem to be that that was the case.
Tim, a few slides back, when you showed your reduction, right there, perfect. It seems that maybe the deep layers are somewhat spared versus superficial, or is that an optical illusion to me?
I mean, it didn't reach significance, so yeah, it's possible. I mean, during development, you make these deeper layers first, and then the more superficial layers later. So it's possible that if we think there's changes in cell cycle dynamics, that's going to occur over time. So the cells that are most likely to be affected are the superficial ones, the ones born later. So there is a biological developmental reason for thinking that superficial cells could be have a greater effect in this mutation than the deep ones. Yes. Okay, so the other thing we wanted to explore was the changes in the gene expression profile in this mutant mouse. So to do this, we perform multiome analysis. So in this assay, you harvest the cells and you obtain both the single cell RNA and the single cell chromatin accessibility from the same cells. So we have all that information integrated here. We did it in the 3 different genotypes and the 2 different time points. And again, this is a UMAP plot. So in this case, we've integrated the RNA and the chromatin accessibility together, kind of in this, it's called weighted nearest neighbor. You can do the same type of analysis. You can look at the expression profile of genes that map nicely into pseudotime. So up here, you have your ventricular zone cells, and as you start to branch out, you get post-mitotic cells. What we were really excited about is that, again, in this mutant, we see more somatostatin cells in the adult. Somatostatin is expressed in the embryo, and what you can see in the knockout, we see a pretty significant increase in the number of somatostatin cells in the knockout compared to the wild type. So that's consistent and fits with the changes we saw in cell fate. Conversely, while PV is not expressed at this age, there's 2 genes that have been associated with parvalbumin expressing cells, MEF2C and MAF, are both enriched in what I'd call PV fated cells. And what you can see here is that both of these genes, you see much higher enrichment expression of these genes in the wild type, especially D15, than you do in the knockout. And again, this kind of fits with what we saw in the adult mouse. And because this is multiome data, we can also look at chromatin accessibility of these motifs. And you can see that these motifs are significantly more located at accessible regions, much more in the wild type compared to the knockout. And kind of a more unbiased approach, you can just compare genes, chromatin accessibility profiles enriched in wild type and knockout. And you see down here D15, again, the 2 genes that are enriched in PV fated cells. We see them enriched in wild types. Somatostatin is over here and a number other of genes of interest. So it looks like this readout we see in cell fate changes in the adult mouse brain seems to be a rise from these changes in gene expression early on in the MGE. Now, the last thing we did is we were interested if these changes in methylation, if they occur sort of evenly throughout the genome, or if they're kind of hotspots, regions where genes are maybe more susceptible or more resistant to the change, to the modification. So in this case, we're performing cut and tag to look at the K27 methylation in the wild type and knockout. I'll just note the way we did this analysis is that we kind of ignore that global reduction that we saw in the mutant. We're looking at relative differences between K27 trimethyl at various genes. And what was really interesting is that we did see some genes, here's FOXP1, which was significant at both ages, where in the knockout, there's just an incredibly strong relative reduction of K27 trimethyl in the 3 trials we did compared to the wild type. Conversely, we see other genomic loci where here at NKase 2.1, you see that there's actually a slight increase, or I should say relative increase of K27 methylation at the NKase 2.1 locus in the knockout compared to wild type. So we think this could possibly apply to other regions where there might be genomic loci that are more susceptible to epigenetic perturbations, in this case FOXP4, or ones that are more resistant, in this case at a gene that's basically the master regulator of MG cell fate. And so we think it'll be interesting in other models to explore how broad changes in epigenetic perturbation affect different genes differentially, some being more susceptible and some being more resistant. Okay, so that kind of wraps up our story looking at what happens when you affect gene repression. Now, in our ongoing work, we've kind of done the opposite to see what happens if you take a sledgehammer to kind of block or perturb gene activation in the MGE. And so gene activation, one critical aspect of it is methylation at histone 3 lysine 4, H3K4. This is really critical for promoter activation. I'll note that dysregulation of this methylation is associated with a variety of neurological and psychiatric diseases. And then this really nice summary slide that looks at mutations in various genes in relation to human syndrome. 3 of them have mutations in genes that directly are involved in methylation of this residue. So understanding this is really critical both for normal development and its possible role in various diseases. Now, the problem is that unlike EZH2 and K27 methylation, there's 6 different family members that can methylate K4. So just knocking out one gene could be problematic because you could have compensation from other family members that can still methylate this residue. So we were lucky that our colleague at NIH had developed a Cre-dependent mouse line where they mutated this lysine residue to a methionine, so H3K4M, and basically this disrupts methylation at this residue and thus severely perturbs promoter activation. And so this is just the genetics. We've crossed this mouse with NKX 2.1 to specifically target MGE cells. And I'll just note that since this is a transgenic in the homozygous mice, you have 2 wild-type copies of the histone allele and 2 mutant copies of the histone allele. So conceptually, it's like a functional heterozygote. It makes it possibly a useful study to think about its relation to disease. So the first thing that was pretty evident from these mice is that the homozygous were smaller than their wild-type and het littermates, both in terms of body size as well as in terms of brain size. And it's a little bit more evident here with a cross-section with a number of these homozygous mice. You saw an expansion of the lateral ventricles that we never really see in the wild-type or het mice, indicating probably shrunken cortex or shrunken regions of the brain and expansion of the ventricles.
Well, such widespread effects for the Cre-dependence should just be MGE.
Yeah, so technically MG hypothalamus preoptic area. So I say it's not MG specific, but yeah, it's basically going to affect the cells down here. But again, you have a ton of, well, not here because this is a thalamus, but you have a ton of structures down here in the ventral forebrain, the striatum, globus pallidus that all contain MGE derived cells and that are going to be affected. So you're affecting interneurons up here, and you're affecting also ventral structures down here, as well as some cells in the striatum. So
You kind of meant more body size, or you just think that's secondary to failure to thrive?
nope. So I'll show you why we think that is in a second. So in addition to the changes in brain mass, we also saw that about 20% of the homozygous mice died in about 3 weeks. And to Greg's point, what we were surprised to see is that for the mice that survive, they eventually catch up and bypass their litter mates in terms of size. They catch up by about 4 months, and by about 9 months, they're literally double the size of the wild types in the heads.
Well, so is that an issue here with the phenotype?
Yeah, so there's 4 regions where NKX 2.1 is strongly expressed: the MG, the hypothalamus, the thyroid, and the lung. And so we have a whole nother project looking at NKX 2.1 function in that, but almost 100% that the reason we see these changes in weight is due to NKX 2.1 Cre mutating gene expression in the hypothalamus and or thyroid. This is certainly not MGE.
I was thinking of thyroid because of.
Yeah. So it's really important for thyroid. And again, this result in combination with kind of my general curiosity of what NKX 2.1 is doing in general regions is why we're kind of studying NKX 2.1's functions in these different organs right now. So back to the interneurons, what we see, again, staining in the adult mice, we see again a slight reduction in the overall number of interneurons. But in this case, we see it occurs, this reduction is present in both somatostatin cells and parvalbumin cells. So in the EZH2 mouse, we kind of saw a shift in fate. One type goes up, one type goes down. In this case, there's kind of a general downregulation of all types of MG-derived interneurons. And we're currently doing the counts in the hippocampus as well, but it looks like this effect might even be more severe in the hippocampus. And those counts are ongoing. Another thing we noticed in these mice is that they seem to be susceptible to seizures. Here's a homozygous mouse undergoing these rapid forelimb movements that are indicative of seizures, and you can see wild type and het littermates around here. So this didn't happen in all the homozygous mice, but it certainly was noticeable. We also saw other homozygous mice like this. This one's a little bit more smaller and wobbly, but you can see it undergoing kind of a tonic-clonic seizure where it basically freezes. And then if you let this video go on, it basically doesn't move for about a minute. So obviously there's perturbations in interneuron function here. We put these mice through a seizure susceptibility assay by injecting PTZ. And what we can see is that the homozygous mice have a significantly higher susceptibility for seizures. In fact, we actually had to drop the PTZ concentrations much lower than what was on my protocol because the homozygous mice died within 5 minutes of injection. So they're extremely sensitive to this, and that's not observed in the wild type or the het mice. So they kind of motivated us to put them through a battery of other assays. They kind of look like they had locomotor defects. So we've been assessing their anxiety, their locomotion, learning and memory because of the apparent defects in interneurons in the hippocampus. And I'll just show some of this data. Again, this is still all ongoing and pretty early. But if we look at the open field test where you put the mice in an open field, I think it's quite visually striking. You can see that the homozygous mice basically avoid the middle area, but they're also running at about twice the speed and twice the distance. So these mice are just zooming around the edge of this box in the same amount of time. And this is indicative of high anxiety in a novel environment. Another type of anxiety assay is the light-dark box. You just have a box, 50% dark, 50% light, drop the mouse in, and you can see that the homozygous mice spend a lot less time in the light zone, another indicator that they're more anxious than their wild type and het littermates. We've also seen some other types of behaviors if you just monitor their mice in the home cage. So we have this system where you can just drop the home cage in and then look at the amount of times the mouse breaks the laser beam over a span of 4 days. We see significant decrease in general locomotion of the mouse in their home cage in the homozygous mice. We noticed as we were handling these mice, they were very jumpy. In addition to having seizures, they would run around the cage when you handled them. Sometimes they would just jump out of the cage, and it was actually kind of difficult. So we put them on this cliff avoidance where you put a mouse up here and you figure out how long it takes the mouse to jump off. Wild type mice do it very rarely. The homozygous mice are happy to take the plunge and jump off the cliff. And lastly, we started to do some working memory tests. So the Barnes maze is where you have all these circles. There's one target where the mouse can crawl into this hole and kind of be safe on a ledge underneath. And so the homozygous make much more, spend a lot longer to find that hole, more error time or time in these error zones compared to the wild type. So we think there could also be learning and memory perturbations in these mice as well. So behavior is great. It tells us something, but again, I'm a molecular biologist, geneticist, so I really want to know what's different in this mouse model in terms of the gene expression epigenetic profiles. So basically, we perform multiome assay on both the MG and hypothalamus of the wild type and the K4M mice. So we've had this data for about a month, so this is all quite preliminary. Again, the UMAP plots in green is the MG, and red and pink is the hypothalamus. We see putative cell clusters. We can look at gene expression profiles. But what was cool, so if you look at the ventricular zone cells up here, and then the postmitotic cells are down here, if you focus on the MG, the green, you can kind of see a salt and pepper organization here in the ventricular cells where the wild type and the homozygous cells are overlapping. But as the cells differentiate, you can see quite distinct streams as if when the cells are becoming postmitotic, they're turning on different transcriptional profiles. And that makes sense because there should be massive perturbation of gene activation here. So that's the case both in the MG and in the hypothalamus. They're almost completely separate. And so this is what we're currently investigating what genes are differentially on and off. And just to highlight, we do see a couple of genes enriched in the MG or the hypothalamus in these wild type or homozygous mouse. And again, there's a ton of data here for us to explore. Okay, so what I've kind of told you about today is that we've been able to establish this epigenome atlas, which is, in our opinion, define the ground truth of cells in these distinct regions of the embryonic mouse brain. Published that in Nature Communications. The RNA data was in eLife, and then kind of our protocols to generate all this data, how we do single cell and single nuclei dissociations from adult and embryonic mouse brain. I've told you about this story with EZH2 to looking at how gene repression changes cell fate and maturation, which is currently out on bioRxiv, and the story about modifying gene activation with the K4M mutation is currently ongoing. And again, these were kind of two sledgehammer approaches. What we want to do in the future is focus on genes that are more directly related to disease, one of which is ARCS, which is a transcription factor strongly enriched in interneurons, in particular the MGE. Mutation of ARCS is associated with intellectual disability. So we've imported floxed ARCS mice that we're currently crossing with NK-Cre and various reporters to perform similar types of assays in a mouse mutant that better replicates human disease. So I hope I've kind of showed you how we're trying to make progress, understanding how you go from this heterogeneous region in the embryonic mouse brain to generate this incredible diversity of GABAergic inhibitory interneurons, both in terms of normal development and possibly to gain a better understanding of neurodevelopmental diseases. This couldn't have been done without all the lab members I've had, everybody highlighted in yellow, current lab members, a variety of very talented postdocs and postbacks, all of the support, in particular from course at NICHD, and the funding from the NIH. Thank you. I'm happy to answer any questions.
Can you hear me now? All right.
Go bring him the mic.
Okay. My question is about K4M knockout mouse. Based on your change, you showed the somatostatin cells decreased, right? It looks like for me, it's more like the deep layer, not superficially, right?
Based
on your picture, looks like.
Yeah, so I don't have that data here. We did.
You show one image. Yeah, this image. It looks like for me,
so I believe that's correct. We did, when we did the counts, we separate superficial and dorsal. I didn't put it here for simplicity. I believe that's correct, but I would have to go back and check that data visually. I think you're right. It does look like there might be differences in layers. If I remember right, it's significant for both superficial and deep, but it might have been a stronger effect in the deep.
Your quantification also show the PV cell also changed. This is the total number or the density? Is it density, right?
This is density. We've also done like the percentage like we did in the EZH2, but this is cells per square millimeter. So this is density. So density of both PV and SST are going down, as is the overall number.
Do you think another possibility is this, how you explained this decrease? Is it because of the migration less or because of the cell death apoptosis? What age is this about?
So this is P21. So this would be after the programmed apoptosis that occurs in interneurons P7 to P10, something like that. So yeah.
Do you think which is the reason? Is the migration less or because of the apoptosis?
Yeah, so it's a good question. This is all a snap in time of the adult. So this doesn't have any mechanistic insight. My best guess is because gene expression patterns are so messed up early on, as these cells are differentiating, my guess is that it's due to changes in neurogenesis. It could be migration. If it was migration defect, which you sometimes see in migration defects, is a buildup of cells in various regions. The cells don't migrate properly up into the cortex.
Yes.
We didn't really see that in this mouse mutant. We don't see a bolus of red cells like stuck trying to get up to the cortex. So I don't think there's a massive change in migration. My best bet is perturbation of normal neurogenic processes because of these changes in expression, but it's something we have to explore. We don't know yet the mechanism.
So my last question is, do you observe the PV cell, somatostatin cell, their morphology changed? Because it's the last mutant, you see some change. Do you observe this morphology change or not?
Those are ongoing. Literally Tuesday, the last 3 days, our colleagues in Chris McBain lab have been recording these cells. So they probably have 60 plus, well, probably more than that, 60 to 80 plus cells that they've recorded the last 3 days that they will process, hopefully take a break for the holidays, and probably get back to me in January with changes in physiology and then ideally changes in structure. So those experiments are ongoing.
Hi, great talk. If you can go to the volcanoes for the repression project, there were like MEC3 and Rion, which were, I guess, downregulated. And yeah,
This one?
so in the E12.5 plot, they are downregulated in the knockout, right? If I'm
Yeah.
correct. So what do you think of the PRC2 complex is doing? Is it by chance that they're more susceptible to the mutation, or there's a transcriptional program in place that regulates this region, MEC3 Rion, that happens to regulate the neurogenesis in the?
Yeah, so one thing I have no good explanation for is why we see such a significant difference between E15 and E12. You can see lots of genes enriched in wild type or mutant here, and then at E15 and E12, we really don't see a huge amount of differences. And I didn't go through some of the single cell data, but that seems to be the case as well in the RNA-seq. If we look at differential genes, I mean, I kind of did hint at that. Here, most of the differences we see are at E15 and less so at E12. So NKX 2.1 starts to be expressed at E9.5, E10 in the MGE. And so it takes time for the CRE to be expressed and then the recombination to occur. My best guess is because we think these changes are happening over time with changes in cell cycle dynamics. You just might not have as strong an effect of knockout of EZH2 changing the overall behavior of these cells at E12 than you do 3 days later when these cells have undergone additional cell cycle dynamics and there's a greater exacerbation. Another way to think about it is maybe loss of this gene is having a 10% effect in gene expression or changes. So that's very minor at E12, but 3 days later, 10% with, if it's about 3 cell divisions per day, another 9 cell divisions, you can start to see greater changes in gene expression. But in terms of why you see only enrichment here and knockout of these, some genes really strong compared to wild type, I don't have a good explanation for that.
Thank you.
I was interested in in the beginning you showed in the human, the diversity of the interneurons was expanded to an even greater extent than in the mouse. I was wondering, like, are those mainly MGE, CGA, LGE, and have you in your data have you looked at mouse versus human to see what those differences might be?
Yeah, so it is color coded. It's hard to see over here, but the purple is going to be MGE and the orange. So that's SST. So sorry, this is SST and that's PV. So the orange and red here is MGE and the purple here is CGE. So in terms of differences between mouse and human and interneurons, what has been shown is that there's a significant and you can kind of see it here. There's actually a significant expansion of CGE cells in human compared to mouse. There's a study came out last year that did EM for mouse somatosensory cortex and human somatosensory cortex. And basically there was about a 4-fold increase in overall interneuron density. So more interneurons in humans compared to mouse, but in particular CGE derived cells. So they call them bipolar in the paper because it's all EM. So bipolar most likely means VIP cells, which come from the CG. They saw an 8 to 10-fold enrichment of these bipolar interneurons in the human cortex compared to the mouse. So I think there's both an expansion in the overall percentage of interneurons in the human compared to the mouse, as well as quite possibly differential diversity, or there's certainly increased subtypes of these interneurons in the human compared to the mouse.
Yeah, I have two simple questions. So the first question is, in the first part, so when you knock out the EZH2 in the user NKX 2.1 cree,
Yep.
so the neuron seems like EFE character is similar, right? Not is normal. I
Yep.
just wondering, is any behavior change?
So they didn't seem to have spontaneous seizures, and we didn't proceed with doing any behavior testing because we didn't see any evidence of them having disrupted behavior in the cage. But we didn't put them through the battery of assays we're doing with the K4M, in part because we weren't motivated to, because we didn't see any striking behavioral phenotypes like we did in the K4M. So my guess is no, but we didn't really look at it.
So that's another simple question is, so is the K4M mutation identified in the seizure patient?
In seizure patients, epilepsy patients? That's a good question. It's more been associated with, broadly speaking, with kind of neurodevelopmental diseases. Intellectual disability is very broad. I don't know the specific details, but it's quite possible that some of these have epilepsy. Same thing here. They just have intellectual disability. So to be honest, I'd have to go and look into these syndromes in a little bit more detail to know how often or what percentage of them have this defect. Again, the mouse model is great and useful for us, but it doesn't exactly mimic a human disease. There's as you can see here, the diseases arise from mutations in the genes that methylate the histone. In our mouse, we've mutated the histone itself. These genes presumably are functional in the mouse. So we think of it as kind of being able to look at how dosage effect of methylation can lead to these diseases. But certainly, autism spectrum disorder, schizophrenia, these have memory defects which we see in the mice. And so we think there are, relations. We're trying to figure out if we want to do any kind of drug addiction in the mice, since perturbation of H3K4 is associated with that, but we haven't figured out if I want to spend the time to write an IACUC protocol to do that yet. But
you were talking about long distance promoter to promoter interaction, or whatever, region interaction. What is the upstream mechanism that determines that? Because it looks like because of that promoter interaction, the fate of neurons will be affected. And then what is it, simply accessibility of chromatin, or is there some more active process? For example, if there is external and external signal from neighboring cells, which determines the differentiation pathway, how will it be linked to those long distance chromatin interactions?
Yeah, so that's a good question. I certainly don't think it's passive. I think it's active. You have various transcription factors or other machinery binding to a region, an active promoter, and how exactly other promoters recognize that or come together to have a large region of transcription isn't well understood, at least not by me. I'm not an epigeneticist by training. There's people that really study this in stem cells or in vitro that might have greater insight. But I certainly don't think it's sufficient to just have two regions to be accessible for them to come together. It's a much more active, dynamic region, a dynamic interaction. And we can see that even from some of our data. We can see regions that are accessible, and sometimes there's a high C contact in some cells, and sometimes they're still accessible in other regions, but they don't have that high C interaction. So I think there's much more mechanistically going on that we don't understand.
So I have a question. So in general, there are distinctions between the removal of repression through histone marks and removal of activation, particularly in cancer. And
Yep.
if I remember correctly, removal of repression is actually more consistent with
Yeah.
transformation. So in your EZH2 mice, did you see any indication that there was rapid progenitor proliferation, even some, intermediate that may have gotten?
Yeah, what the literature seems to support is that loss of EZH2 tends to lead to exit of the cell cycle. And we think, although we didn't directly test that, we think that's what's going on in our mouse, that we're depleting the progenitor pool too early. I didn't say it, but PV cells tend to be born later, and somatostatin cells tend to be born earlier. So if in this mutant you're having forced neurogenesis too early, you would make more somatostatin cells early and have fewer cells to make parvalbumin cells late. So we think there's a
So the removal of repression in this instance, which you would assume generally would lead to anomalous gene expression.
Yeah.
things that is actually in this case driving neurogenic.
it seems that perturbation of EZH2 in general, not just our study, but others, seems to kick cells out of the cell cycle.
So
too
in other
early.
parts of the nervous system as well.
So there was, I think the other study was in the cerebellum or maybe the midbrain, where they looked at that more directly than we did, and they saw loss of progenitor cell, making postmitotic cells too early or depleting the progenitor pool. However you want to think about it, yeah.
Great. Other questions? Online questions? We don't, anything in the chat? All right. Well, thank you so much, Tim, for a
Okay.
great seminar.
Thank you.